Hello guys, welcome back to my channel. It's Dr. SNK. So coming to the new subject, that is microbiology. So what is basically microbiology? It is the lab diagnosis of the infectious diseases. Any of the investigation you're going to do for the infectious diseases, that is inculcated as a branch of microbiology. So basically, coming to the exam pattern, in the first half I'll be covering regarding the general microbiology, immunology and few part of the system such as CVS. Okay, so next coming to the second half, I'll be covering the respiratory system, all those things. When it comes to the hospital acquired infections, I'll be dealing it in the first half. So basically first half you have more portions. First half will be divided into part 1 and part 2, whereas second part will be published in a single video. So today I am going to discuss the first part of the general microbiology and immunology and few systems. Okay, in the second part, here I will be dealing with general microbiology and immunology. So if you like the video, please like, share and subscribe my channel and stay tuned for the more. So starting with general microbiology. In the general microbiology, there is something called as dark ground microscopy. Okay, dark ground microscopy. It is a type of microscope where it is used to identify very small organisms. So what is the keyword here? Very small. Okay, very small organisms are visualized under this microscope under a dark background. Okay, so how are they made visible? They are made visible by adding silver impregnation. So what are the organisms you are going to see under dark ground microscopy? First is Treponema pallidum, which is a causative agent of syphilis. Next it is Borrelia. Borrelia will cause relapsing fever. Okay. Next, coming to the Leptospira. So basically what is Leptospira going to do? Leptospira is going to cause leptospirosis. So in the leptospirosis, what are the things you're going to concentrate there? How are they seen basically? Treponema, it is slender and flexible. Spirally coiled. Okay, slender, flexible and spirally coiled. Next, Leptospira, how is it? Leptospira is basically tightly and regularly coiled. Okay, you're going to see the regular twists and turns. Okay, regular twists and turns. So if you're going to draw the diagram, how is the diagram there? You're going to draw it tightly coiled. That is, it is tightly and regularly coiled. Tightly and regularly coiled is Treponema. Whereas, tightly and regularly coiled is Leptospira, whereas Treponema, it is bit slender and flexible. So you're going to see the hooks here. Basically in the Leptospira, it is hooked in the edges. Next, Campylobacter. So Campylobacter, it specifically exhibits the darting motility. That is what you're going to appreciate there. Next, coming to the organisms identification, that is using silver impregnation. You are going to impregnate the bacteria with silver so that the bacteria will be visible under the dark ground as a white bacteria and it is enlarged in size. Okay, because they are very small, you are going to impregnate them. You are going to add extra things. That extra thing is silver here. Coming to the electron microscope. So what is electron microscope? It is basically, it is used to identify the smallest structures visible. In the entire India, there are only few centers which are having this particular electron microscope. One is Tata Memorial in Mumbai and most probably AIIMS Delhi. These are the two things which are the institutes which will be dealing with electron microscope. So basically very few laboratories have this and it is highly costly and the machine itself is very large. It occupies maximum space. So that will be added as a disadvantage. Okay, if they are asking the disadvantage, it is expensive, it is space occupying and it is not affordable by the lower people as it is not cost effective and it is not easily available and it requires a trained personnel. All these things are the disadvantages. So what is the use of this particular electron microscope? It is used to identify the ultra structure. I told, right, very small structures, that is ultra structure, that is identified by the microscope, that is by electron. Using electron beam you are going to identify the organisms, ultra structures. And you are going to see the cell organelles also, such as mitochondria, Golgi complex, lysosomes. All these things are visualized under electron microscope. Next, it is used as a part of quality control to check the quality of the product, as a part of failure analysis. Why this product has failed, for what reason did this particular thing failed, that is called as quality control. To identify that minor things or minute mistakes in the project what you have done, you are going to use the electron microscope. Then it is used to produce electron micrographs. Okay, there is something called as electron micrograph that is prepared by this electron microscope. Next, to study microorganisms to effective the treatment. Okay, you are going to study the microorganisms in detail so that you can give the best treatment possible for the patient for that particular microorganisms, which will be effective. Next, weaving of the frozen section or the frozen material. If you want to weave any frozen thing or the frozen material as a part of forensic examination, you have seen so many movies. They are going to freeze many specimens and they are going to re-establish them and see. And it is seen most commonly in the electron microscope because it is highly precise and effective for diagnosis. Next, 3D tissue imaging. You are going to make the 3D tissue imaging. You are going to convert the tissue into a 3D structure. That is possible by electron microscope. Next, coming to bacterial cell wall. So this is the 5 marker. Please concentrate. It is very important also as a part of university exams. Cell wall of the bacteria. So what is cell wall basically? Cell wall is a tough rigid structure. Okay, remember this. It is a tough rigid structure which is surrounding the bacteria. Next, what is it made up of mainly? It is mainly made up of peptidoglycan. It is mainly made up of peptidoglycan. Is the main component. So what is basically this peptidoglycan containing? It contains some residues or the repeats called as NAG and NAM. Okay, NAG and NAM. That is N-acetylglutamine and N-acetylmethionine. These are the two things you are going to, N-acetylglycine and N-acetylmethionine. Next, coming to the gram positive and gram negative. How we are going to differentiate them? So basically, first I have written gram positive is having thick cell wall, whereas gram negative is having thin cell wall. So how are we going to differentiate them now? Since gram positive is having more of peptidoglycan layer, it is thicker. It is having thick layer, whereas gram negative it is having very less peptidoglycan layer formed. So it is having thin bacterial cell wall. Next, what is the composition basically? Here you are going to see the pentaglycine bridge. You are going to see the pentaglycine bridge is present here itself. Here it is absent. Next, the lipid content. Lipid, where is it present more? In case of gram negative. Why I will tell you. Remember the next point, that is lipo. Okay, that is lipid. Lipopolysaccharide is seen more in case of gram negative. That's why the lipid is more present in case of gram negative. Next, teichoic acid. What is teichoic acid? It is a peculiar thing which is present in the body with various functions I am going to discuss in the near future. It is present only in case of gram positive. It is present only in case of gram positive. Next, coming to the functions of the cell wall. Functions of cell wall. Functions of cell wall is protection of cell content. It is going to protect the cell contents. Next, it maintains the shape of the cell. So you should be in this shape only, bacteria. Okay, this cell of the bacteria should be in this particular structure, that is maintaining the shapes. It maintains the shape. It protects the cell contents. Next, it maintains what? Osmotic integrity. Okay, it maintains osmotic integrity. Next, it helps in attaching to other cells. It helps in attaching to other cells. Then not only it is helping in attachment to the other cells, but also it is helping in attachment to the macrophages. It helps in attachment site as a part of macrophage. Help in role of cell division. It will help in cell division. Then it is a platform for flagella, villi and base and fimbriae. it lacked like a basement membrane as a support or a paya or a so much things which have been used as a stone of support. Okay, for whom? For flagella, pili and fimbriae. Okay. Next, coming to the function of teichoic acid, it helps in antigen determinant. Okay, it helps in antigen determinant. Then it regulates the normal cell division. Then it is a source of magnesium. It is a source of magnesium. Coming to lipopolysaccharide. Okay, lipopolysaccharide, and what are its composition, or what is its composition, or what are its components basically. So LPS is having lipid A and core polysaccharide and O antigen. These are the three things which are present in case of lipopolysaccharide. So if they are going to ask what does the lipopolysaccharides contain, it contains lipid A, core polysaccharide and O antigen. So what is the function of this lipid A? It will recognize the bacteria. So what is the function of core polysaccharide? It will produce endotoxin. So what is the function of O antigen? It reacts with the antibodies. Next, coming to the bacterial spores. In the bacterial spores, how are you going to define spores? Basically, spores are the dormant forms of bacteria which are thick-walled and resistant to so many things in the surroundings. That is what. Okay, they are the dormant forms. They are thick and they are resistant. These are the things you are going to mention in a definition. So basically, what are the bacteria which produces the spores there? So there are two types basically, mainly, that is bulging spores and non-bulging spores. Bulging spores example is Clostridium. So it is going to cross that particular cell wall out. It is going to cross that particular bacterial cell wall. Okay, it is coming out. That is called as bulging spore. That is seen in case of Clostridium. Next, non-bulging spores, you are going to see the bacillus. Okay, the spore is present within the cell wall. That is non-bulging. Whereas based on position, it is having various types. That is terminal. It is present at the end. That is seen in tetani, Clostridium tetani. Then subterminal, just near to the end. That is seen in case of Clostridium welchi. That is involved in case of forensic. Okay, to bring about that last process called as liposear mummification. Okay, after death, this is that bacteria, anaerobic bacteria, which acts on our body. Then central spore. Okay, central spore, that is seen in Bacillus anthracis. So what I have studied, spores studied now, that is terminal, subterminal, central. These are all oval. Next, coming to the spherical, there is only one spherical, that is seen in case of tertium. Okay, Clostridium tertium. Okay. Next, coming to the properties of spores. It is extremely resistant. Okay, they are resistant. Then it is non-reproducing. Then it is non-stainable. You are not able to stain that particular spore. Then it is metabolically inactive. It is not able to do any of the metabolism inside it. That is metabolically inactive. And it has the capacity to germinate after long period. After long period, this particular spore, it can become a future cell after process called germination. So how we are going to draw the spore diagram now? So spore diagram, basically this is how we are going to draw. That is outer layer. Okay, complete outer layer is called as exosporium. Okay, the outer layer, the fifth most layer, that is called as exosporium. Fifth layer is exosporium. Then come inside. After coming, you are going to see the spore now directly. You are going to see the spore. Exosporium is just outside the spore, so it's called as exospore. Then fourth one, it is spore. Fourth is spore. Okay, fourth structure is spore. Then third structure is, what is the third structure? That is cortex. Okay, cortex is present in the middle. Remember like that. Then next it is core. Okay, core membrane, which is enclosing the core. Then it is last, you are going to see the core. Okay, core is covered by core membrane. Spore is covered by exosporium. In the center, you are going to see the cortex. Okay, this is how simply you are going to remember. Then uses of spore. Where are they used? Basically spores are used in case of sterilization control. In case of sterilization control you are going to see the spores. Then to deliver biomolecules. Then last, they act like a vehicle for vaccines. Then they can be used as a bioterrorism agent. I should not aggravate it because bioterrorism is not good. So these particular things they are used as a bioterrorism agents. Then killing of spores. Basically, how are the spores killed? Basically, there are only two methods which are effective against spores. That is high level disinfection and chemical sterilization. These are the only two things where the spores can be killed. Okay, next coming to the bacterial growth curve. So this is what the bacterial growth curve. We are going to plot it as the time passes. What is the changes seen? So this is time. Okay, this is time. This is the changes in cell. Okay, changes in cell, and this is the time. So basically first phase is called as lag phase because there is no increase in anything, so it is called as lag phase. Then you are going to see the growth, that is called as log phase or exponential phase. Then you are going to see the stationary phase. There is no growth here again, stationary. Then you are going to it is seen in the decline, that is called as decline phase. So we will deal in detail what are the things here. Coming to the lag phase. So what is lag phase? What are the characteristics you are going to see to compare these phases? One is division, okay, that is number division or the cell number division. Here there is no division, here it is no division, that's why it is in lag phase. Then is there any cell death here or the bacterial death? No, there is no cell death or the bacterial death. What is the total count here? It is flat because there is nothing happening. Viable count is again flat. What is viable count? Viable count is the bacteria which are alive. They are called as viable bacteria or the viable bacteria. That is called as viable count, whereas total count is all the bacteria including the dead things. Okay, next log phase. So in the log phase basically you are going to increase the cell number, so the cell division is maximum here, or it has the maximum potential. Okay, there is so much of cell division that the cell is starting growing so badly. Okay, let's continue. Then bacterial cell, is there any cell death here? No. If the cell death is happening, the curve should go down. It is not going down, it is keep on increasing, so there is no death here. Then total count is increasing irrespective. Viable count is also increasing because the new bacteria are being formed. Next, coming to the stationary thing, there you are going to see what? Cell division. Yes, right, you are going to see the cell division, but what is the difference between them here now? What is the difference between them now? The bacterial cell death will start occurring here. Remember, the bacteria will start dying slowly. So the total count is increasing because irrespective death and live bacteria are there. So that is what total count. Viable bacteria is flat now. Why? Because the death is increasing. Then decline phase, what are you going to see? You are going to see the cell division? No. The graph is coming down. See here, the graph is coming down, so there is no cell division. Then bacterial death, yes, bacterial death is maximum here. What is the total count now? Total count is becoming flat. Why? Because the bacteria cells have already started dying now. So because of that reason, you are going to see this change, that is flat change. Next, viable count has started decreasing. Viable count has started decreasing. So what are the unique features which is going to happen in each phases? That's what I've written here down. Okay, these are the unique things. So basically in the lag phase you're going to see so much of enzymes and metabolites because of no cell division. Enzymes and metabolites are being accumulated so that in the future the cells will start doing their part. Next, in the log phase, you're going to see cells uniformly stained and active. They are active now because they are actively dividing. That's why there is log phase, that is exponential. It is growing, exponential growth, or the log phase or the log growth. Then it is stationary. Stationary, what is stationary? Basically, there are again here producing so much of granules. Okay, granules. You're going to so much of granules. Then exotoxins, spores, antibiotics, all these are the things which are being concentrated in this particular stationary phase. Then coming to the decline phase, you're going to see the involution forms. You're going to see the involution forms. Okay. Then bacterial capsule. So how are you going to define the capsule? Capsule is a large amorphous viscid material lying outside the cell wall. Okay. It is amorphous and viscid. Okay. Amorphous and viscid material which is being seen just outside the cell wall. Then what are the bacteria which are having this particular capsule, capsulated organisms? One is Pseudomonas. Pseudomonas. Then is Pneumococcus. Then is Meningococcus. Then is Cryptococcus. So many cocci are present here. Remember, pneumococci, meningococci, cryptococci. Okay, pneumococcus, meningococcus, or cryptococcus. Okay, all these are the cocci. Then it is Pseudomonas and H influenza, that is Haemophilus influenza. Then Klebsiella and Bacillus. Okay, these are the things. So next, functions. What is the function of this particular capsule outside there? What is the function? It will inhibit the phagocytosis. Okay, then it is a source of vaccines and nutrients. It can act like a source for vaccines and nutrients. Then it forms biofilm. It helps in formation of biofilm. So it will act like a mechanism of resistance for the bacteria for few antibiotics because of formation of this biofilm. Then coming to the staining procedures, basically how we are going to stain this capsule, there is something called as negative staining. Here the capsule is not going to be stained, that's why it's called as negative staining. That is done by India Ink, and you are going to see the cryptococcosis here. Basically cryptococcosis is most commonly seen in this way. Then coming to McFadden staining. Okay, McFadden staining, you need not know any procedures of this. Okay, McFadden staining, you are going to see the Bacillus anthracis. McFadden is Bacillus anthracis. Then it's Quellens reaction. Quellens reaction, you are going to see the pneumococcus. Then next you are going to see what? So was it visible? Sorry, was it visible for you people? Yeah, it was visible. Okay. Then flagella. What are these flagella? Flagella are thread-like appendages. Okay, they are thread-like appendages. They are thread-like appendages which contain a protein called as flagellin. Flagellin, simple. It is containing a protein called as flagellin. So what are the parts of this particular flagella? It has three parts. One is the filament, then it has the hook and the basal body. Filament, hook and the basal body. So how are we going to represent it on bacteria? If suppose this is a bacterial cell wall, what happens? This is the filament outside lying. The one which is attaching, the one which is curving, that is called as hook. The one which is covering, that is called as hook. The one which is attaching to the body of the cell wall, that is called as basal body. Okay, this is basal body, this curved part is hook, and this long filament structure is called as filament. Simple, filamentous. It is very thin and long, that's why it's called as filament. Then coming to the organs. What is the function of this particular organs? Now it helps in locomotion or motility, to move the bacteria. These things are helping them. Then types, there are types called as mono, loco, peri and amphi. Okay, mono. Okay, how I used to remember is MLA. Okay, MLA. Okay, there is someone called as Prakash and he is a MLA now. Okay, Prakash is MLA. Or Periswami, simple, Peri is there, so Periswami. So Peri MLA means big in Tamil. Peri means big, so big MLA. Okay, the small structures, in spite of they being small, they are going to do the big job, that is called as locomotion. So peri MLA. Then mono, P is for peritrichous. Okay, M is for monotrichous. L is for Locotrichus. A is for Amphitrichus. Amphi means present everywhere. Okay, peri means in the periphery. Mono means single. Loco means everywhere. Okay. So what are the examples under them now? For monotrichus it is Vibrio cholerae. For Locotrichus it is Pyralium bacteria. Then peritrichus it is Salmonella typhi. Then staining. How are you going to stain this particular flagella? You are going to visualize this particular flagella by dark ground. Okay, I told it right. Very small structures you are going to see under a dark field microscopy. In a dark background you are going to impregnate with the silver stains. Okay. Then phase contrast and electron microscopes. What are the motilities? Basically, what are the types of motilities you are going to focus here now? What are the types of motilities? Tumbling motility, which you are going to see in Listeria. Swarming motility you are going to see in Proteus. Darting motility you are going to see it in Cholera. Corkscrew like motility you are going to see in Spirochetes. Then coming to the culture media. It is a 5-marker and it is an important question. As a part in general microbiology, you are going to focus here properly. So what is a media? Culture media. It is a media containing substance that has nutrients. Okay, it has nutrients that help in the growth of microorganisms. Okay, that help in the growth of microorganisms such as bacteria and fungi. So what are the basic components of this media? Peptone and agar. Okay, peptone and agar. So what are the types? Basically, what are the types of this cultural media? What are the types of cultural media? Simple media or basal media. Simple or basal. So here you are going to see the growth of all non-fastidious organisms. All non-fastidious organisms grow here. So what is the component of this particular thing? Here you are going to draw what? Extra, that is nutrient broth. Okay. Here I told it for every media what is the basic? Peptone water and the nutrient agar. Agar should be common. The extra here is nutrient broth. So it is used for the base for preparation of all others media, and it is used to do tests such as catalase tests, oxidase tests. All these tests are done under this simple media or basal media. Coming to the enriched media. Enriched media, it is containing both basal media plus what? Blood, serum and egg. Okay, blood, serum and egg. Remember, it is used only in case of fastidious organisms. Then what are the examples of this particular enriched media? It is blood agar, okay, chocolate agar, low-flush serum slope, okay, and blood culture media. These are the examples which come under the enriched media. So what is the function of this blood agar? It is helping in to see the hemolysis. Hemolysis is seen. Hemolytic property is tested here. Coming to the chocolate agar, it is a selective media for H. influenza growth. Okay, low-flush serum soap, it is used for Corynebacterium diphtheria. Next, blood culture media. Okay, it is two types, mono and biphasic. Monophasic and biphasic. In case of monophasic, you are just going to see BHI. What is the full form of this BHI? Brain heart infusion. Repeated, brain heart infusion broth. You're just going to see the broth here. Next, in the biphasic, agar is added. You're going to see the agar. Okay, you're going to see the agar. Coming to the types of cultures media, as explained previously, basal media is done, enriched media is done. Now we are going to move on to the enrichment media. Okay, enrichment media, we are going to enhance. So it is used for selective organisms growth and decrease the normal flora. Okay, for selective organism growth, and it decreases the normal flora growth. So selenite F broth. Okay, selenite F broth and alkaline peptone water are the two examples under them. Selenite F broth, it is used in case of Salmonella and Shigella, whereas alkaline peptone water, it is used in case of Vibrio cholerae. Then coming to the selective media, it is same as a enrichment media, that is, it increases the growth of selective organisms, whereas it inhibits the growth of normal flora. So what are the examples here? Selective media, it is very selective for that particular organism. So LJ media, it is specific for Mycobacterium tuberculosis, TCBS. Okay, you're going to Google all these full forms. Okay, TCBS media, it is Vibrio species, for all the Vibrio species. Then potassium tellurate agar, PTA, potassium tellurate agar, it is used for Corynebacterium diphtheria. Then XLD and DCA, these are very specific for Salmonella and Shigella. If the specimen is from stool, remember it should be from stool. Okay, it should be from stool. Then coming to next, that is WBBS media, that is Wilson Blair bismuth sulfate media, that is used for Salmonella typhi. Then transport media, it is used for transport. Okay, it is used to transport delicate organisms which are very delicate, which cannot be sustaining the outer environment. Then what are the examples here? One is Pike's media, that is used for Streptococcus. Then Stewart's media for Neisseria. Venkataramanan's media and Clary Bear media, it is used for Vibrio cholerae, which is the most commonly asked. And this Clary Bear media, it is also used for Shigella and Salmonella transport. Then one more thing, that is buffered glycerol saline, that is used for again for Shigella and Salmonella. Coming to differential media, they help in differentiating the groups of bacteria. That's why they are called as differential media. So what are the examples coming under them? It is McConkey agar, then Cled agar. These are the two things you must differentiate. These are the two things, examples you need to remember in case of differential media. So McConkey agar, how is it helping? It is going to help in formation of lactose fermenting and non-lactose fermenting. If you are going to see the pinkish colonies, they are called as lactose fermenting. And what are the examples under them? E. coli and Klebsiella. Non-lactose fermenting, Shigella and Salmonella. Cled agar is again same as above, that is McConkey agar, but what? It is used for urine samples. Remember, it is used for urine samples. So what are the extra advantages of this particular Cled agar? It is less inhibitory for other organisms and support the growth of few other bacteria. So okay, so what are the advantages? It helps the other bacteria also to grow, except what? Except there are two bacteria, that is beta hemolytic streptococcus and candida, are not allowed for growth. Okay, then what is other function? It is inhibiting the swarming. Swarming is produced by basically protease. Okay, protease. It is also a type of swarming motility, which I have explained in the previous slide. Okay, then coming to the anaerobic cultural media, what is it? It is not containing oxygen, that's why it's called as anaerobic. Okay, it reduces the oxygen by adding the reducing agents. Okay, how? By containing the reducing agents in its media, it is going to reduce the oxygen content, thereby making the bacteria to grow in the absence of oxygen. Such bacteria are called as anaerobic bacteria. So what are the mediums which are used? Basically, what is an organism used in this? It is Clostridium. Okay, Clostridium is the most common encountered anaerobic bacteria. So what are the medias here? Robertson cooked meat broth, RCM broth. Then it is thioglycolate broth. Then brain heart infusion sulfate. These are the anaerobic medias. Okay, this is what about the cultural media. Then coming to Albert staining. What is Albert staining? Albert staining, it is very specific to stain the metachromatic granules. Okay, to see the metachromatic granules which are being produced by the Corynebacterium diphtheria. Also, these bodies are called as polar bodies or volutin granules. Okay, polar bodies or volutin granules. Composition of this particular Albert stain, it is containing two stains, that is toluidine blue plus melachite green. It is called as Albert A stain. If it is Albert B, it contains iodine, whereas others, Albert A contains toluidine blue plus malachite green. What are the methods of gene transfer? Next question, that is methods of gene transfer. There are three methods. One is transformation, the other one is transduction, and the last one is conjugation. So basically in transformation, what is it? The process of random uptake of a free DNA fragment from surrounding media by bacterial cell wall and incorporating it into a chromosome. Okay, there is a transformation, there is a change. Okay, that is called as transformation. Process of a random uptake of a free DNA fragment from surrounding media by the bacterial cell wall and incorporating into the chromosome of itself. There will be some free DNA roaming just around the bacterial cell wall. It is going to be taken by the bacterial cell wall and it has been incorporated by various receptors and it is been introduced or incorporated into the chromosome. That is called as transformation. Coming to transduction. What is transduction? Transformation of the DNA portion from one bacteria to other by bacteriophage. Okay, by bacteriophage. Here the bacteriophage is helping. What are the types under transduction? There are two types. One is generalized, the other one is localized. Generalized and localized or specialized. Sorry, not generalized and specialized, not localized, specialized. Then what are the uses of this particular transduction? It is used as a part of drug resistance and as a part of treatment. Conjugation, transfer of the genetic material from one to another by mating with each other. They are going to do the mating process with each other. The one cell will mate with the other cell to form this particular process, that is called as conjugation with each other. Okay, both cells, they are going to mate with each other and there is a formation of conjugation tube which is in between them. What are the types? F plus into F minus mating, then HFR conjugation, then F dash. These are the three things you are going to remember. Plasmids. So what are plasmids? How we are going to define the plasmids? They are the extra chromosomal double stranded circular DNA. They are extra chromosome, they are double stranded, they are circular DNA. Okay, it is extra chromosomal double stranded circular DNA that exists in free state in cytoplasm of the bacteria. Where do they stay? They stay in the cytoplasm. They stay in the cytoplasm. What are the types of plasmid? Types of plasmids are F plasmid, that is fertility plasmid, which helps the sex pili and formation of the conjugation tube as a part of conjugation. Then R plasmid, that is helping in the resistance. Okay, resistance and antibiotics. Then coal plasmid. Coal plasmids, they contain what? They contain bacteriocins. Then virulence plasmids. Virulence plasmids, they help in virulence, that is toxins and adhesins. Okay, these are the things which have been coded by these plasmids. Then it is metabolic plasmid, which help in the metabolic activities. Then vectors. Okay, they are vectors. Where they are vectors in case of genetic engineering in the formation of recombinant DNA technology. Remember your PUC thing. There you are going to see the plasmid which are being incorporated into the gene of desirity. Okay, basically what is formed? It is used in protein production and gene therapy. Protein production and gene therapy. F plasmid, R plasmid, coal plasmid, virulence plasmid, metabolic plasmid. F plasmid for fertility, R plasmid for resistance, coal plasmid for bacteriocins, virulence plasmid for toxins and adhesins, metabolic for metabolic activities. Vectors in genetic engineering, that is recombinant DNA technology to produce a protein and in gene therapy. Coming to the modes of transmission. What are the modes of transmission? There are direct and indirect. Direct is by direct contact. You are going to touch the person directly, that is direct contact. Then it is droplet infection. Then contact with soil. You are going to touch the mud or soil. Then inoculation. Inoculation into skin. Okay, inoculation into skin. That particular soil what you touch, you are going to inoculate it into the skin. Then vertical. From mother to baby. Then indirect. In the indirect you are going to see vehicle borne, vector borne and air borne. Okay, there is some one mediating this, that's why it's called as indirect. Coming to the bacterial classification. So in the bacterial classification, one is gram positive, other one is gram negative. Gram positive cocci, gram positive bacilli. Okay, in the gram positive cocci, it is staphylococcus and streptococcus. Then in the bacilli of the gram positive, that is bacillus, clostridium and corynebacterium. These are the things. Then coming to the gram negative cocci and bacilli. Gram negative cocci, gram negative bacilli. Gram negative cocci, one example is Neisseria. Gram negative bacilli, E. coli, Shigella, Salmonella. Okay, these are the things I'm going to write it. Then coming to the basics now. There are so many basics you're going to do. First you're going to do general biology, then you're going to do the general parasitology part, then you're going to do the general mycology part. Okay, these are the things done. Then first we are going to start with viral replication. So because the viruses do not divide by a normal method, that is binary fission, as it is seen in bacteria, there is a separate process called as viral replication involving various steps. Okay, how are you going to remind this? How are you going to remind the order of steps? There is one girl called as Asma. You're going to tell it. Okay, you're going to tell to your friend, at pub Asma re. You're going to use re, be and all for your friends, right? Re, o, be, kidhar chal rahe, wo bol rahe na, wo sab things you're going to see. Okay, you're going to say it for your friend. At pub Asma re, there is some girl called as Asma re at that pub. Okay, at pub Asma re, there is some girl as Asma. Okay, remember her, and you're going to tell your friend re, and where is she? At pub. So first is at is for attachment. At is for attachment. The receptor interaction occurs between the virus and the host. Then penetration. Okay, penetration. So what are the subtypes you're going to see in penetration? One is phagocytosis, that is via receptor mediator the virus is going to enter inside via a process called as endocytosis. Then membrane fusion of the membranes of the virus and the host, that is seen in case of HIV. Then injection of the nucleic acid. Injection of the nucleic acid is done by whom? There should be someone, right? That is done by bacteriophage. Then coming to the next process, at is done, P is done. Now next is pub. In pub, U is for uncoating. So uncoating, basically the capsid and the nucleic acids are released now. Okay, the capsid and the nucleic acids are released out of that virus. Now it is coming out of that inner structure of the virus. It is going to come out, that is called as uncoating. It is opening its coat, that is called as uncoating. This particular step is absent in case of bacteriophages. Then coming to biosynthesis. In the biosynthesis, it is used in case of synthesis of DNA or RNA. Okay, DNA or RNA basically in its genome. Then capsid and enzymes. This is a formation of whom you are going to form the nucleic acids, capsid and enzymes. All these are formed. That is called as biosynthesis. It is going to increase its material, that is DNA or RNA with capsid and enzymes. So biosynthesis, what are the sites? Basically all the DNA viruses, you are going to see the biosynthesis in nucleus, except pox, you are going to see it in cytoplasm. Then all the RNA viruses, cytoplasm, except retrovirus, orthomyxovirus. Okay, retrovirus and orthomyxoviruses, it happens in nucleus. Coming to the next step, asthma. Now that girl, asthma, A-S for assembly. Okay, asthma, A-S for assembly. Who is going to assembly there now? It is forming all its materials, right? That is DNA or RNA with capsid and enzymes. It is going to be formed in single place. They all assemble in a single place. That particular process is called as assembly. So here the viral nucleic acid with proteins, they are going to form the viruses now. That is called as nucleocapsid. In DNA, all the assembly will occur in the nucleus except hepadenovirus, where you are going to see most commonly hepatitis B virus, then pox virus. Then RNA, it is going to happen in cytoplasm. Coming to the asthma, M-A for maturation, asthma. Now that girl name is ended, asthma is ended now. Maturation takes place where? It happens in nucleus or cytoplasm membrane. Then release, last ray for release. Release of what? The doctor virions, that new things are formed, right? They are going to mature in the maturation phase, where it happens in the nucleus and the cytoplasm or the membrane. Then they have been released out. No, those daughter virions are released out by what? By lysis of the host cell. That happens in non-enveloped viruses and bacteriophage. The outer layer of the cell is being lysed and the inner contents come out of it. Then budding, it happens via budding through host cell membrane. Host cell membrane, there is a budding formed that is happening in enveloped viruses. Then there is something called as eclipse phase. You are going to remember in this viral replication. Basically what this phase represents is the viruses which is in the host. From then on you are going to calculate the period till the appearance of the first virus particle formed. That phase of empticity where the virus is in the host and its first progeny is been appearing in the body. That difference between other phase, it is called as eclipse phase. Then what are the abnormal replicative cycles you are going to see? Incomplete viruses, okay, incomplete viruses are formed, then defective viruses are formed, then pseudo virions, okay, then abortive infections. These are the things which are going to happen: incomplete, defective, pseudovirions and abortive infections. Then coming to viral cultivation, how are these viruses cultivated? Usually they are intracellular, so they require some cell inside for their growth. They cannot be grown on a normal cultural media as a part of bacteria and fungi. Okay, they grow easily on the bacterial media, that is the cultural media, whereas virals, they don't grow. So viruses do not grow on artificially cell-free media. They don't grow, okay, they doesn't grow on a artificial cell-free media. What are the types? One is animal inoculation. You are going to inoculate the virus into the animal. So what are the most commonly used animals? That is suckling mice. Okay, suckling mice is most commonly used. So if you are going to inject this suckling mice being injected by the bacteria, most commonly Coxsackie virus and arboviruses are tested here. So Coxsackie A virus, it is going to develop the flaccid paralysis, whereas Coxsackie B will cause spastic paralysis. Flaccid means loose or very labile. B is spastic, very hard. Remember. Then second is embryonated egg. You are going to introduce the virus into the embryonated egg. So what are the locations inside an egg? One is chorioallantoic membrane. Chorioallantoic membrane, that is used as a part of pox formation in case of vaccinia and variola and HSV 1 and 2. Whereas yolk sac is for Japanese encephalitis, rickettsia, chlamydia and Haemophilus ducreyi. It is very difficult to mug up these things, so there are no mnemonics for them also. If you are creating the mnemonics, indirectly you will end up in confusing all the mnemonics at the end in the exam, and you are going to remember the mnemonic, but you are not able to answer. To remember that mnemonic, you are going to create some other mnemonic, again you go and make the blunder there. So it's better avoid useless mnemonics. Then amniotic membrane for influenza. Then allantoic cavity, it is used for influenza again with yellow fever and rabies. Then coming to the tissue culture. Tissue culture, basically you are going to use it in an organ, you are going to use it in explant and cell lines. These are the three varieties of tissue culture. For organ, it is used in the coronavirus for tracheal ring. Tracheal rings of the trachea are used for the culturing of coronaviruses, whereas the adenoid explant, okay, you are going to take out the adenoid and from there you are going to use the adenovirus cultivation. And in the cell lines there are three. One is primary, the next one is secondary, the last one is continuous. Okay, primary, secondary and continuous. Primary, secondary and continuous. In primary you are going to use the rhesus kidney cell line and chick embryo fibroblast. Here we are going to use the chick embryo fibroblast, whereas in secondary you are going to use the human fibroblast. And if you are going to remember, you can remember them, that is MRC5 and WI38. These are the secondary cell lines, whereas in continuous it is very simple: HeLa Hep2, Vero McCoy, HeLa Hep2 Vero McCoy, HeLa Hep2 Vero McCoy, HeLa Hep2 Vero McCoy. These are the continuous cell lines. Coming to the explanation of each cell lines now, that is important. Primary, it is having limited divisions, okay, limited divisions. It can only used for limited divisions of the bacteria, that is rhesus kidney. It is used for mixo and adeno. Both of these organisms, they are going to cause pneumonia, okay, or the common cold. Myxoviruses contain orthomyxo and paramyxoviruses, okay, that is influenza viruses, and adenoviruses. Both of these, they are involving the upper respiratory tract infection. That is how you are going to remember. Then secondary is having moderate divisions, okay, moderate divisions. In the moderate divisions, you are going to see the human fibroblast, that is used for CMV, and it is used for preparation of viral vaccines, okay, viral vaccines preparations also. This particular secondary cell lines are used. Then coming to the continuous, if they are dividing continuously, remember what might they be? They will be cancerous cells. They will be using cancerous cells here. Indefinite divisions, indefinite divisions. So what are the cell lines? Hep B, HeLa, McCoy and Vero. So Hep B, what is it used for? Hep B is used for, Hep 2 is used for respiratory syncytial virus, adenovirus and herpes simplex viruses. Then McCoy is used for chlamydia. Vero is used for rabies vaccine. Simple. Then detection of viral growth. How are you going to determine whether the virus is growing in that particular culture? There is different forms of things. One is cytopathic effect. It is a 3 marks question, remember and write it properly. What is cytopathic effect? How you define it? It is a morphological change produced by the virus in the cell line detected by the microscope. So there is some morphological change which is produced by the virus where in the cell line, which is detected by whom? By the microscope. So what are the different types of the cytopathic effect? One is formation of syncytium. Okay, there is a group of cells which are formed syncytium. That is seen in case of measles and respiratory syncytial virus. Whereas the second one, it is cell necrosis. You are going to see the cell necrosis and lysis. That is seen in case of enterovirus. Then cellular clumping. You are going to see the cells clumping with each other. That is seen in case of adenoviruses. What are the other things? What are the other cytopathic effects? Rounding, okay, rounding of cells and discrete focal degeneration. I am not able to do the diagrams there, which are very complicated and it is not sufficient for you people to remember all those things. So better ignore. So these are the things you are going to concentrate here. Then next method, you are going to see the virus growth rate. How? It is by viral interference. What is interference? You are going to intervene something now. So growth of non-cytopathic effect virus when challenged with cytopathic virus. Okay, if there is already a non-cytopathic virus inside, you are going to see the inhibition of the cytopathic effect. Okay, suppose example rubella being a non-cytopathic effect virus, it is going to be causing the inhibition of the CP virus, that is cytopathogenic viruses, that is enterovirus. Okay, if there is already a non-CPE growth, it is going to be inhibited the cytopathic effect of the new growth. Then to detect antigens, okay, antigen detection, that is done by direct immunofluorescence, then immunoperoxidase staging and hemadsorption. If possible, you write it, or else just write to detect the viral antigens. Then electron microscopes. In the electron microscopes, you are going to see the viral particles directly. Okay, viral particles are seen directly. Then viral gene detection. Viral gene detection is done by PCR. Then coming to the inclusion bodies. So in the inclusion bodies, what are the things seen? Basically, one is aggregates of virions, are the viral particles and other products of viral replications which are being demonstrated on the light microscope. So what are the composition of inclusion bodies? Virions, okay, are the viral particles in simple. Then its products, okay, which are formed as a part of viral replication, which is being detected by the light microscopy. Virions or viral particles with its products. Virus plus its products form the inclusion bodies, which are being seen under light microscope. So what are the types of inclusion bodies? Intracytoplasmic, intranuclear, and both. So intracytoplasmic, basically Negri bodies, that is seen in rabies. Then molluscum bodies, that is seen in the disease name itself, that is molluscum contagiosum. Then Paschan bodies, that is seen in variola. Intranuclear, there are Cowdry A and Cowdry B. In the Cowdry A, there are two, that is Torres bodies, which you are going to see in yellow fever, and Lipkels bodies, you are going to see it in HSV, herpes simplex virus. Cowdry B is specific for polio. Cowdry B is specific for polio. Both intracytoplasmic and intranuclear, you are going to see it in measles and CMV. In CMV, that appearance is called as owl's eye appearance, which you are going to differentiate from Hodgkin's lymphoma, which looks very similar. Coming to the general parasitology. In the general parasitology, they are going to ask you about the protozoal infections and their classification. So I simplified it like this. So what are protozoa? Protozoa are the microscopic eukaryotic. Okay, they are microscopic eukaryotic, can be free living. They can be free living or parasitic. Okay, they can be free living or parasitic. And they can be heterotrophs. What are heterotrophs? They feed on the other organisms for their nutrition. Okay, other organisms. They feed it on other organisms. Then what are the forms of these protozoans? One is cyst, other one is trophozoite. What is cyst form? Cyst form is both infective form and diagnostic form. What does it mean by infective form? Infective form means this particular cyst is going to cause the infection. Then diagnostic form. What is diagnostic form? Seeing the cysts in the microscopy, you are going to say that this particular infection is this. So it will work as diagnostic form also. So if you are going to see the cysts, it is indicating both infection and the diagnosis of that particular disease. Are cysts motile? No, they are non-motile. Whereas trophozoite, it is again can be motile or non-motile. So basically seeing on this, trophozoite is basically a diagnostic form. So based on these diagnostic forms, you are going to classify as ciliates, which will contain cilia as their locomotion organ. Then amoeba containing pseudopodia as a locomotion organ. Then flagellate, the name itself tells flagella as a locomotion organ. Whereas sporozoa, they are non-motile. Remember, sporozoa, spore. Okay, they are non-motile. Okay, they are non-motile. Coming to the examples, seeing the examples here, ciliates, basically ciliates, Balantidium coli, okay, Balantidium coli, that will cause gastroenteritis, that is going to cause the gastroenteritis. Whereas amoeba, it is having two forms, intestinal form and free living form. Intestinal form, you are going to see the entamoeba histolytica. Free living forms, Naegleria and acanthamoeba. Whereas flagellate, again intestinal form, you are going to see Giardia, and blood or tissue forms, that is Leishmania and Trypanosoma. Then vaginal flagellate, there is only one, Trichomonas vaginalis. Sporozoa, that is non-motile, again it has two forms, intestinal form, blood or tissue form. Intestinal form, there are Cryptosporidium, Cyclosporidium and Cystoisosporidium. That is Cryptospora, Cyclospora, Cystoisospora. Blood or tissue as a part of sporozoa is Plasmodium, Babesia and Toxoplasma. All these are the parts seen. Coming to the intestinal protozoa. In the intestinal protozoa, ciliates, amoeba, flagellate and sporozoa, all the subclasses contain the intestinal protozoa. So what does this Balantidium coli and entamoeba histolytica cause? It is going to affect the large intestine. If large intestine is involved, then it will cause dysentery. Remember, what is dysentery? Dysentery is blood or mucus, okay, mucus plug, blood or mucus plug with diarrhea. Okay, you are going to see extra blood or the mucus plugs within the loose stools. Whereas diarrhea is just loose stools. You are not going to see the blood or the mucus plug that is caused when it is involving the small intestine. and it is done by the flagellate and the sporozoa. In the flagellate it is giardia, and in the sporozoa all the spora, that is if there is any word containing spora, they are going to cause the small intestine infection and that will lead to diarrhea. The sporozoa contain all the spora things, that is cryptospora, cyclospora and cystoisospora. Then coming to the free living, in the free living you are going to see amoeba. So basically free living you are going to see amoeba. What are the two things you are going to concentrate there? That is Naegleria and Acanthamoeba. Naegleria will cause primary amoebic meningoencephalitis, which is always acute in nature, whereas contrast is Acanthamoeba, which will cause chronic amoebic encephalitis. So usually they will give the history of swimming in the clear water such as swimming pool for these infections, because they are free living and they desire the neat waters, that is the clean waters. Sorry for the disturbance. Then blood and tissue protozoa. For the blood and tissue protozoa, again you have two, that is flagellates and sporozoa. In the flagellates, leishmania will cause leishmaniasis, whereas trypanosoma will cause trypanosomiasis. Trypanosomiasis, there are two types, that is Trypanoma cruzi and Trypanoma brucei. Okay, Trypanoma brucei and cruzi I am going to explain in parasitology. Okay, don't concentrate more here. Then sporozoa. In the sporozoa it is plasmodium and babesia and toxoplasma. So they are going to ask the vector here, please concentrate. Sorry, in the leishmania the vector for the leishmaniasis is sandfly, okay, or phlebotomonas. Okay, phlebotomonas. Then coming to sporozoa, it is plasmodium, babesia and toxoplasma. Plasmodium will cause malaria, babesia will cause babesiosis, toxoplasma will cause toxoplasmosis. Okay, simple. The name itself tells all. Then vaginal, I told you there is only one vaginal, that is trichomonas, and that will cause trichomoniasis. Typically they will give the history of green vaginal discharge. If they are going to say the vaginal discharge is green in color, just mark your answers blindly as trichomoniasis, which is caused by Trichomonas vaginalis. And you are going to see the whiff test positive here. If you are going to see the clue cells, that is seen in case of bacterial vaginitis. Okay, that is it is caused by bacterial Gardnerella vaginalis bacteria. Mosquito has vectors. Come now, next, mosquito has vectors. Where? Aedes. Come on, tell me. Aedes, it is used in case, it is as working like a vector in dengue, chikungunya and yellow fever. Anopheles in case of malaria. Anopheles will cause in malaria, whereas Culex in Japanese encephalitis and filariasis. Then skin penetration parasites. Skin penetration parasites are basically trematodes and nematodes. Okay, trematodes and nematodes. These are the classification of helminths. Okay, cystodes, trematodes and nematodes are the three classifications of helminthology. Whereas in parasitology, that is protozoa, I have told ciliates, flagellates, amoeba and sporozoa. Whereas in case of helminthology, cystodes, trematodes and nematodes. In trematodes, cystosoma is skin penetrating. And in nematodes it is strongyloidosis and ankylostoma. Okay, what are the skin penetrating? Did you see them? Yes, you have seen it. Then coming to the peripheral smear parasites. Which are the parasites you are going to see on preparation of the peripheral smear in the patho department? So malaria, babesia, both of these are seen in blood. Okay, being in a blood and tissue parasites, you are going to see malaria and babesia. Then you are going to see the trypanosoma, then brugia, Mansonella and Wuchereria. All these are the peripheral smear seen parasites. Then coming to the stool concentration techniques. Okay, stool concentration techniques. In the stool concentration techniques, what are these basically stool concentration is used for? Stool concentration is a technique which is used mainly perform to separate the parasites from the fecal debris. Okay, it will separate the parasites from the fecal debris. So its number is increased. Okay, on the sedimentation, increase in the number is seen after this process. And what does it do? It will unmask whom? It will unmask the parasite and makes them visible so that we can study the parasite. Then what are the types of stool concentration? One is flotation technique, the other one is sedimentation technique. In case of flotation techniques, they'll add the increased specific gravity solution so that these all the ova and cysts, they are floated on the top. That's why this particular medium is called as, particular technique is called as flotation technique. Whereas sedimentation, you are going to sediment the parasite. So you are going to add whom? You are going to add the formalin. What will this formalin do? It will extract the fecus materials from there and sediment the parasite where? In the bottom after centrifugation. So if you are adding the formalin, that formalin is going to extract all the fecal debris and the fecal matters and precipitate whom or sediment whom? The parasite where? At the bottom. After what? Centrifugation. Then sedimentation. In the sedimentation, what are the organs which have been seen? What are the eggs seen? Ankylostoma is seen, egg. Okay, egg of the ankylostoma is seen here. Then entamoeba cyst is seen, whereas fasciole egg is seen. Okay. Coming to the flotation techniques, what you are going to see is isospora cyst. Okay, isospora cyst, sufficient. This is sufficient. Then coming to the non-biled stains egg. Okay, non-biled stained eggs. That is a mnemonic called as Neha. There is some girl called as Neha. Remember Neha. So Necator, N for Necator americans, E for Enterobius formicularis, H for Hymenolapis nana, A for Ankylostoma duodenale. Then which are the things which are going to be floating in the saturated salt solution? Okay, saturated salt solution, which are floating there. Okay, which are going to be floated in saturated salt solution. Same EHA is going to repeat here, that is Enterobius. See, Enterobius. H for Hymenolapis nana, here also Hymenolapis nana. A for Ankylostoma, here also A for Ankylostoma. With them you are going to add his friend, that is Necator. He is always similar to the Ankylostoma. Okay, you always take your friends together, right? So Ankylostoma also likes his friend and takes him together, that is Necator americans. A is for Ankylostoma and extra A here is Ascaris, and T is for Trichura. Here the mnemonic is Teha. Neha, Teha. Remember like that. Here it is Neha, here it is Teha. Then bile stained. Which are the bile stained? There are only two things, that is Ascaris and Trichura. So simple. Neha, okay, plus 80. Okay, Neha plus 80. Neha plus 80 is salt solution. Okay, saturation. So both of these, all these things will be included here. A plus B is equal to C. Same way, this C thing is saturated salt solution. A is containing bile stain, non-bile stain, B is containing bile stain. Together all this, they are going to float in the saturated salt solution. Then what are the cystodes which are causing infection in the man? One is Taenia, Solium and Saginata. Okay, Taenia, Solium and Taenia, Saginata. Okay, Taenia, Solium, basically it is causing what? Cysticercosis. If it enters into the brain, it is called as neurocysticercosis. Its preform is called as cysticercosis cellulae. Okay, this is the preform. It is causing teniasis, whereas Echinococcus granulosus will cause echinococcus, that is hydrated cyst, and Diphyllobothrium latum in the HIV patients it will cause diarrhea. Okay, whereas H. nana is the other one, that is Hymenolapis nana. These are the cystodes which are infecting the man. So what are the operculated eggs? Which are the operculated eggs? You are going to see cap for eggs, that is called as operculum. Like cap it is formed. Okay, this particular thing, that is cap for an egg, there is some cap, that is operculum. So what are the eggs there? You are going to see Fasciola hepatica, Fasciola busci and Chronorchis and Paramysmus westernmani. Okay, all these are what? All these are what? Trematodes. Remember, all these are trematodes. Repeat them. These are all trematodes. Extra one is Diphyllobothrium, that is a cystode. Remember, Diphyllobothrium is the only cystode which is operculated, whereas all the operculated eggs, if you're going to see a cap over the egg or the operculum, you're going to see all the trematodes except womb, that is cystosoma. Okay, cystosoma is not seen. Except cystosoma, all the other trematodes, that is Fasciola hepatica, Fasciola busci, Clonorchis and Paranismus westernmani, you are going to see. Only cystode having operculum is Diphyllobothrium latum. Okay, coming to the general mycology. What is in the general mycology? First important topic is dimorphic fungi. What is dimorphic fungi? In the name itself, it has two morphological forms. That is why it's called as dimorphic fungi. It has two forms, that is hyphal form and yeast form. Hyphal form is this flat structure. Okay, you're going to see the flat structure like this. If it is having the septations, it is called as septate hyphae. If it doesn't have any septations, it's called as aseptate hyphae. Simple. Then yeast form. In the yeast form, you're going to see this yeast-like organisms. So basically, these hyphal forms, they're going to see it in the culture, whereas yeast forms you're going to see in the sample. In the sample itself, you're going to see. In the sample, if you're going to do the microscopy, these yeast forms are seen. But it's these hyphal forms, you're going to culture, then only you're going to see the hyphal forms in the microscopy, culture microscopy. The two forms, hyphal form, okay, it is forming in the cold temperature, that is 25 degree Celsius, whereas yeast form in the body temperature, that is 37 degree Celsius. So compare now, what are the dimorphic fungi? Histoplasma, coccidiomycosis, paracoccidiomyces, blastomyces, sporothecschenchi and talaromyces. These are the fungi. Okay, these are the fungi. So histoplasma, this is the yeast form, this is the mold form, that is a hyphal form. So yeast form, it is the only intracellular fungi. So you are going to draw a cell, inside that you are going to see the yeast forms. It is the only intracellular fungi. Then mold. In the mold you are going to see the tuberculate conidia. You are going to see a tuberculate conidia. Coccidiomyces, you are going to see spirules in the yeast forms. Whereas in mold form you are going to see a anthraconidia with fragmentation. Paracoccidiomyces, you are going to see a Mickey Mouse. So what is the mold form in case of Paracoccidiomyces? It is pear-shaped conidia. Then coming to blastomyces. Blastomyces is having broad base, okay, broad base buddings. You're going to see the broad bases, right? So they are very broad here. They are called as broad base budding. Whereas blastomyces, it is very similar for mold form of Paracoccidiomyces. Okay, it is pear shaped. Whereas Sporothecschenchi, it is having asteroid bodies, just like a star asteroid. Then Sporothecschenchi mold form is having petaloid or a flower petal appearance conidia. They are uniformly arranged. Then Talaromyces, or it is also called as Penicillium marnefi. It is having oval things with septa in case of yeast form, whereas mold form is having branching conidiospores. Coming to the opportunistic fungal infections. In the opportunistic fungal infections, how are you going to define it? They are a group of fungal diseases which are present as a part of normal flora, become activated and to form diseases when the immunity is low. They are a part of normal flora. They get activated when there is a low immunity and they form disease. What are the examples? Candidiasis, cryptococcosis, mucormycosis, aspergillosis, penicillosis, fusariosis and pneumocystis. Remember, Candida, Cryptococcus, Mucor, Asper. Candida, Crypto, Mucor, Asper. Penicillium, Fusarium. Okay, Penicillium, Fusarium. Then pneumocystis. So we're going to see in detail this because it's an important 5-mark question for you. Candidiasis, it is caused by a fungal called as Candida, and it is most commonly seen in HIV or AIDS patient. Okay, HIV or AIDS patients. So clinical manifestations where and all is going to be affecting mucosal, cutaneous, invasive and allergic. These are the morphological forms of the clinical manifestations. Lab diagnosis, in the microscopy you are going to see the gram-positive oval budding yeast cells, with pseudo hyphae, Then culture, on the culture you are going to see the creamy white pasty colonies on the Savarots dextrose agar, SDA, Savarots dextrose agar, it is highly specific for fungal growth. Then germ tube test, if it is positive, just blindly mark Candida albicans. Okay, Candida albicans, you are going to see the pseudo hyphae there. You are going to see the pseudo hyphae. There is no constriction at the site of formation of hyphae. That's non-constrictable. Coming to the treatment, if it is cutaneous or the mucosals, you are going to give azoles. If it is disseminated, then amphotericin B. Okay, liposomal amphotericin B. Then cryptococcosis. What are the risk factors here? HIV patients where the CD4 count is less than 200, and immunosuppressive agents and steroid agents. If the people are in these drugs, then cryptococcosis is common in them. What are the clinical manifestations there? Pulmonary, that is pneumonia, and in the meninges, it is meningitis. How are you going to diagnose? I told Cryptococcus capsule is diagnosed by negative staining. You are not able to see the capsule here. The white thing you are going to see it, right? That is the capsule. It is not stained. That's why it's called as negative staining. On the SDA, you are going to see the mucoid creamy colonies. Wherever you are going to see the mucoid colonies, it indicates the bacteria or the fungi, it is capsulated. Then gram stain, you are going to see the gram-positive round budding cells. If it is oval, it is Candida. If it is round, it is Cryptococcus. Then bird seed agar, it is very specific. There you are going to see the brown to black colonies because of deposition of melanin. Then treatment, if with the CNS involvement, if without, sorry, I have written reverse here. Please change it. Okay. If without CNS involvement, it is just fluconazole. If with CNS involvement, then it is amphotericin B, fluconazole, and based on the severity you are going to add the flucytosine also. Okay. Then mucormycosis, what are the risk factors here? DKA patients and iron therapy patients. Okay, DKA patients and iron therapy patients. Coming to the clinical features, what are the clinical features there? Rhinocerebral mucormycosis. Okay, basically rhinitis and sinusitis are being affected, and the cerebrum, that is the brain, that is most common in case of mucormycosis. That will cause orbital cellulitis, proptosis, and diminution of vision, and even sometimes the loss of vision is also seen. On the light microscopy, you are going to see a broad septate hyphae with obtuse angle branching. It is septate, remember, it is septate hyphae. Coming to the LPCB, that is lactophenol cotton blue. It is a special stain to demonstrate the spores, and the spores seen here are sporangiosspores. These are the spores what I have drawn. If you are going to see rhizoids, they are not seen here. Okay, basically rhizoids are absent. Remember, rhizoids are absent. This is how we are going to draw. Okay, with the sporangiospores inside here, whatever the dots I have formed, that is sporangiophore inside, and it has come out. Okay, this is how we are going to draw the mucor fungi. Coming to the other things, that is aspergillosis. It is common in case of diabetic patients. Remember, it is common in case of diabetes patients. Whereas, what are the clinical manifestations of aspergillosis? It is pulmonary, that is pneumonia, and invasive sinusitis. There are three forms: Aspergillus fumigatus, Aspergillus flavor, and Aspergillus niger. So Aspergillus fumigatus, in the microscopy you are going to see 1/3 conical spores, okay, 1/3 conical spores. Then ascospores, they are called as ascospores, they are sexual spores. Then Aspergillus flavus, that is having 2/3, okay, 2/3 or globular. Okay, then Aspergillus niger, it is having entire, okay, entire, and it is called as vesicular, just like a vesicle full. Microscopy, how we are going to see the colonies now. How are the colonies seen basically? If it is smoky green, if it is very dark green or the smoky green, it is fumigatus, it is having fumes, smoke, fume, remember like that. Then flavors, it is having yellow, okay, flavors. Niger means black. If it is having mild infection, you are going to give itraconazole. If it is having invasive or severe infection, then you are going to give voriconazole. For all the rest things, you are going to give liposomal amphotericin B. Then pneumocystis. In the pneumocystis, it is common in case of HIV in individuals with CD4 count less than 150. Then pneumonia is the main thing caused by this, and the diagnosis is wet mount and the GMS stain, that is gomorimethamine staining, GMS. In the wet mount, you are going to see the cyst, and on the gomorimethamine stain, you are going to see the pink ball appearance, okay, crushed pink ball appearance, which look black cysts, which are crushed, and it is non-cultivable. Remember, it is non-cultivable. Treatment, cotrimoxazole. Treatment is cotrimoxazole. If it is associated with HIV, you are going to give it as 21-day regimen. If it is not associated, if it is 14 days. Remember all these days. If you remember, write them, or else just leave it. Just write the treatment, cotrimoxazole. Deep mycosis. What are the deep mycosis? Examples: mycetoma, which will cause the mycetomal infection, that is Madura food containing the granules. Whereas pyrotrichosis, they are going to give the gardening history, okay, gardening history with subcutaneous nodules. You are going to suspect whom? Sporotrichosis. It is called as rose gardener's disease or rose gardener's syndrome. Then chromoblastomycosis, then pheohypomycosis. These are the 4 things you are going to remember as a part of deep mycosis. Coming to the systemic mycosis. It is histoplasmosis, blastomycosis, coccidiomycosis, and paracoccidiomycosis. Paracoccidio. Repeat: paracoccidio. Then LPCB. What is LPCB? It is lactophenol cotton blue. So what is the function of this LPCB mount? It is going to demonstrate the spores. So what are the spores seen here? Conidiospores, which are of 2 types, that is micro and macro. Micro is seen in case of trichophyton, macro is seen in case of microsporum and epidermophyton. If it is boat shaped, it is microsporum. If it is clubbing shaped, then it is, if it is clubbed with each other, then it is epidermophyton. All these 3 TME infections, they are part of dermatophytosis. They are going to cause the superficial mycosis, that is superficial skin infection. Then sporangiospores. Sporangiospores are seen in case of mucor. Then ascospores, I have told it is seen in case of aspergillus. Coming to carrier. Carrier is the last part in the first half, that general microbiology. So if you're going to like my video, please like, share and subscribe, okay, never forget it. And please share for other ones who require it. I have concised so smartly that you're going to do the best in microbiology, and I have the confidence, and make sure you're going to utilize this session properly and do it in a best way for your upcoming theory exams, or it may help you for practical also sometimes in some topics. Coming to the carrier. Who is the carrier? How are we going to define the carrier? Carrier is a person who harbors the pathologic okay, who harbors the pathogenic microorganisms without suffering from its ill effects, okay, without suffering from its ill effects. So what are the types of carrier? One is healthy carrier, who has been never suffering a disease from that particular microorganisms. Convalescent is he is recovered from that disease but still continues to harbor that particular microorganisms in his body. Then temporary is for less than 6 months. Paradoxical carrier, acquired pathogen from the other carrier, he is going to acquire the pathogen from some other ones. Then contact, if he is acquiring from the patient, okay, if any other, it is paradoxical. If he is having contact with a patient, then it is called as contact carriers. So what are these carriers used for? Basically, carriers are very important source of infection. They are very important source of infection to spread the disease in the community, So you are going to treat the carriers also for some diseases such as salmonella, where you are going to give the norfloxacin, okay. Typhoidal carriers, urine carriers, fecal carriers, which are very common in case of typhoid infection. So there you are going to give the cotrimoxazole, or you are going to give the norfloxacin or ciprofloxacin, okay, or ampicillin or amoxicillin combined with propionate. All these are given as a part of typhoidal carrier treatment, okay. So this is what about the general microbiology. I'll be continuing the immunology in the next session. So first we have completed the normal general microbiology. So please stay tuned for more, and I'll be uploading the immunity or the immunology chapter very soon. And please like, share and subscribe my video lectures, and send it to the maximum people as much as possible who are very needy, as it is an exam time. And make sure you are having a very happy micro time. Thank you. See you in the next video. Bye.